So of course, everyone did good on the case study because we went over it together. If you didn't do good, it's because you didn't write in complete sentences or you misspelled something. It's pretty basic considering the lab was open book, open computer. I dock points off if you slaughtered sentences and spelling. Okay. All right. Let's see. Get off the screen. There we go. Get off the screen too. There we go. Okay, I think this is where we left off last time, right? I didn't get any farther than that. Looks familiar. Okay. All right, so where we are today is we're still talking about what is your innate defense. I hope I can do this. That doesn't require antibodies. It's that 3-week waiting period that you're waiting for antibodies to be produced, and you're extremely vulnerable. So you have to rely on your innate immunity. We talked about barriers, and now if something made it past the barriers and got in your body, I talked about white blood cells and what they do. And then I was on to natural killer cells that secrete interferon or toxic granules. And now we're talking about, let me get this off the screen. There we go. Mast cells. Okay, mast cells are basically basophils. Oh, let's get a different color here. Now, I don't know if you remember this from the blood physiology reading, but mast cells are basophils and they secrete histamine. For inflammatory response. And I'm going to continue up here so I run out of space. That inflammatory response is what you experience in response to an allergen. An allergen is anything that triggers a mast cell to secrete histamine, and we've all experienced it. So pollen season right now, okay? I'm living on Benadryl, all right? So it causes you to have a runny nose. That's your body's way of getting something out of your respiratory tract from there. It causes your eyes to weep because that's your body's way of flushing out antigens that might have gotten into your eye. It causes increased mucus production in your upper airway so you can clear your throat and get that stuff out of your airway. If you've been stung by something, whatever, that site gets red and hot and swollen, that's an inflammatory reaction. So histamine plays a big role in that. And so the reason we take Benadryl, chlorpheniramine, whatever, I always know the active ingredients. So diphenhydramine is Benadryl and chlorpheniramine is, God, I can't remember what that is, but they're antihistamines. They're trying to block that inflammatory response so you have less of that thing going on. All right. And this can also happen not just to an allergic reaction, but a life-threatening allergic reaction, which is, what's that called? It begins with an A, anaphylaxis. That's histamine. Okay. You have a patient come in that sounds like they're having an anaphylactic reaction. They're the first one coming in. Dr. Pryor got stung by a bug one time when we were out fishing. I don't know if I told this story or not, but I recognized he was having an anaphylactic reaction to it because he had splotchy red stuff all over his face and neck and armpit region. His lips were swollen. His eustachian canals and his ears were swelling up and he couldn't hear very well. So I gave him like 4 Benadryl and rushed him off to the emergency room. And I calling on the way, said, my husband's having an anaphylactic reaction. He went past people with cuts and broken bones and they took him right in. All right. And that's what you would do too, because what can happen in anaphylaxis is your bronchioles constrict. You have severe bronchoconstriction from this histamine that can close off your airways so that you can't breathe. Yeah, we talked about anaphylactic shock where it can drop your blood pressure. A drop in blood pressure is not going to kill you right away, but not being able to breathe is or could kill you right away. So that's why they are the emergency. Get them in because number one, you're going to make sure their airways open. All right. So that's a real thing. This histamine. How many people know someone who has would have an anaphylactic reaction to something like shellfish, peanuts, right? It's very common. Insect stings, bees, you name it. Okay. Okay. So is it relevant to nursing? Yeah. Complement proteins, there's like 20 or 40 of them. All you need to know about this is they lyse bacteria by poking holes in them. They do it chemically. And I'm really not going to talk much about them. I don't know if they do in nursing, but basically if you poke a hole in bacteria, osmosis rules that water is going to rush into the bacteria and they're going to lyse, they're going to explode, all right? Okay, so the histamine response. Mast cells can also, I think, secrete the chemical cries for help, but I'm going to kind of streamline this and not try and throw too much stuff at you. But the chemical cries for help, I want you to know, are coming from white blood cells. Okay, so here is a mast cell, a basophil. Here's an allergen that bound to it, and then it's spewing out all that histamine that causes that massive reaction in your body. It's your body's way of trying to get the allergen out of the portals of entry of your body. All right. How many people are suffering from allergies right now? Yeah, I mean, my voice is pretty hoarse from allergies. Okay, so I'm going to do a stepwise what happens once something makes it into your bloodstream. So I'm going to kind of review what I've already gone over, but you remember having to identify some of these cells in lab? Yeah, right, that's a neutrophil, that is a monocyte, that's a lymphocyte, and the other things are red blood cells. So moving this thing, it's always in my way. Okay, so neutrophils, they're the first responders. They're there on site for any kind of possible infection. Monocytes also are in the bloodstream, so never let monkeys. So they're fairly common, not as frequent as neutrophils. But I explained that they're phagocytes. They'll actually consume a pathogen by endocytosis and then destroy it. Okay, but if they're in the bloodstream, they can only act in the bloodstream. Monocytes, I mentioned before, are capable of leaving the bloodstream, and that's what we're going to talk about now. All right, so they destroy pathogens by phagocytosis. They secrete endogenous pyrogens to cause a fever response. And we talked about having a low fever is okay. All right, as long as a person is functional, can eat, drink, and they're not just laid out on the couch unresponsive. Someone pushing 103 and above, that's something you've got to do something about. Okay, so the histamine is secreted by mast cells or basophils. Basophils are a white blood cell. Never let, that's the most infrequent cell. Never let monkeys eat bananas. That's the lowest frequency. Now, this is important. The anaphylactic shock that we talked about, I think we covered on the last exam, is the histamine. It causes all the inflammation that I just talked about, but it also causes your blood vessels to become leaky. All right, and it causes vasodilation. That is the drop in blood pressure with anaphylactic shock is because of this right here. So you're caught between a rock and a hard place. You'll have a systemic wide drop in blood pressure if you're having a major histamine response, but getting the blood vessels to vasodilate and become leaky is the only way that the monocytes are going to escape the circulatory system and get into the tissue. So downside, drop in blood pressure. Upside, you can get those monocytes into the tissue where they can wage war if the bacteria or other pathogens are in the tissue, not the bloodstream. So monocytes escape the bloodstream by extravasation. This is just extravasate. So extra, think outside or out, and vasate, think vessel. Monocytes do that. And then they're going to become a different type of cell that is going to be the beginning of communicating between your innate immunity and your adaptive immunity that's going to involve antibodies. So we're getting close to where this is where you're going to start antibody production. So I have a little GIF here. I hope it shows up. So this is the cells of your blood vessel. And let's see if we can get that to go back. There, here we go. So it's going to slip out of the cells that make up your endothelium of your blood vessel. And the monocyte is going to pop out like that. And now it's in the tissue. And it's called a phagocyte, but it's specifically, it's going to be called a macrophage or macrophage, however you choose to pronounce it. So I'll put here, I don't think I have another, no. So once monocyte escapes blood vessels, I'll say the vessel, it becomes a macrophage. Now, I don't know if you went over any of this terminology in microbiology, but macro means big and phage means eat. It's literally a big eating cell of your immune system that's in the tissue. And when it encounters a pathogen, it's going to consume it, it's going to break it down and destroy it. But it's going to do a little bit more than that. So monocyte becomes a macrophage. So the M becomes another M word. All right, this is just pure vocabulary. Okay, so when that happens, these are going to be the next things that occur. Okay, so monocytes migrate from the blood into the tissues by extravasation and become a macrophage. Here's the word again. That literally means big eater. And it will find pathogens in your tissue and destroy it. But here's the big thing that's going to happen. Once it destroys that pathogen, it's going to take little fragments of that pathogen and it's going to move it to its cell surface. And it's going to be literally holding the antigen out, waiting for a cell of adaptive immunity to come along saying, here is this antigen. I want you to do what you need to do to get antibodies made against this. It's like a waiter out there serving the pathogen's antigen to your immune system. Okay, so the macrophage consumes the pathogen and then it puts little bits of its antigen on its cell surface and it's presenting this antigen to another cell of your immune system. So because it's presenting, it's called an antigen presenting cell. Or APC for short, antigen presenting cell. Now we're at the cusp between internal innate immunity and adaptive long-term and specific immunity. This is the step that's got to happen because the APCs will activate one of the most important cells of your immune system. I gave you lymphocytes, right? And it was even in, I think, the last lab quiz. Lymphocytes include T cells and B cells. Which one of those are actually the most important part of the immune system? T cells, without a doubt, okay? When your T cell count drops below a certain minimal amount per cubic deciliter of blood or something like that, that's where they go, you've had HIV, but now you've switched over into AIDS, acquired immunodeficiency syndrome. And the immunodeficiency is your T cells have dropped too low. Your immune system is no longer functional. It's T cells. So APCs are waiting for a helper T cell, sometimes abbreviated T with a little h. That is the T cells they're looking for. These cells, they don't produce the antibodies, but they do everything to get the antibodies produced. In fact, once the APC communicates with the helper T cell, the helper T cells, okay, I got it now, folks, everyone back off. I'm the director, do what I said. And the helper T cell is going to activate all these other cells and tell B cells to make antibodies. So T cells kind of take over the process from here, saying, I got it, I'm activated, now I'm in charge. And that's literally what they do. So macrophages, not so much helper Ts, but macrophages also send out cries for help with chemicals, okay? So here is the macrophage taking the antigen from the pathogen and putting it on its surface, just like a waiter coming to bring you an antigen, all right? And then it's waiting for a T cell, helper T cell to come along and pick up that antigen and start the process from there, all right? Okay, so here is an APC that's activating a helper T cell. It's like it turns on the T cell and the T cell says, okay, I got it from here. That's it. So it activates it. All right, so continuing on with innate immunity, I just want to introduce the monocyte leaving the blood vessel to become a macrophage, then destroying a pathogen and becoming an APC. Continuing on with other players in innate immunity is the natural killer cells. Yes, I've already talked about it before. God, this thing is annoying. All right. So they respond to the chemical cries for help. They'll release interferon to kill viruses. So I told you how viruses destroy your cells and replicate. They take over your, basically your DNA, your replicating capacity of your cell nucleus, and they cause it to produce new viruses for it, because viruses can't reproduce on their own. They have to hijack your cells to reproduce more virus cells. And when your cells become full of virus, they lyse and then release all these new viruses into your system. Interferon is supposed to slow that down. Okay, and then toxic granules. I know that's a very vague term, but we produce tumor cells quite frequently. And the fact that they don't become cancer is because our immune system is doing things like, I know that you're our tissue, but you're behaving badly. I don't trust you, so I'm going to wipe you out just so that you don't become a problem. So any cells in your body that start replicating and start looking abnormal, your immune system will recognize that, and your natural killer cells will hopefully take care of that before it gets bad. Oop, no, I don't want a quiz. I just want to go on to the next cell. Okay, let's see if this will do something here. Oh, well, for whatever reason, the little GIF is not working, so I'm not going to worry about it. Okay, this is repeat, so you don't have to write anything new, but the mast cells, which are basophils, secrete the histamine for inflammation. Could be just a small local allergic reaction or a huge systemic wide whole body anaphylactic reaction. It depends. But the important thing, it causes that vasodilation to allow monocytes to escape the bloodstream. Again, it comes with the problem of causing your blood pressure to drop. And then complement proteins, they're showing here, here's a rod-shaped bacteria that's got holes being poked into it. And once that happens, basically your osmosis of that bacterial cell is thrown out the window and the cell swells and ruptures. Okay. Sometimes my advanced slide works and sometimes it doesn't. Step by step. I am going to have a question on the final exam. That's why I have a flow diagram. What are the steps from when a bacteria enters your tissue to where your body produces antibodies, and then what those antibodies cause to happen? Step 1, step 2, step 3, step 4, and I'll have them all mixed around. And then the answer choices would be, you have to put the numbers in the correct order, just like I did with the muscle chapter, sequence of a muscle contraction from when a neuron releases acetylcholine to when muscles contract. It's going to be the same type of question. And the reason I do this is because nursing, they have exam questions that are sequential. Because as a nurse, doing things in the correct sequence is important. And you get it wrong, you get the whole question wrong. So I know they test you like that because Dory Weaver has told me. So first step, neutrophils, monocytes are in your bloodstream and they're coming to help, but they are confined to the bloodstream. All right. They secrete endogenous pyrogens to cause a fever response. All right. And so that's the first indication that you have an infection brewing. So of course, the bacteria have breached through your skin barrier and now they're in. So what do these blood cells do? Well, monocytes are going to leave the bloodstream, all right, extravasate and become macrophages. And they become an APC. So once they consume the pathogen, the macrophage, it is now an APC because these things sticking out are going to have some of that pathogen antigen. This cell is waiting for a helper T cell to arrive. It will then activate it, and then we'll talk about what happens after that. Also, responding to the chemical cries for help are mast cells secreting histamine, which allow further vasodilation and leakage of the blood vessels so more macrophages can get in there. All right. And causes all the other things we associate with an allergic reaction. And no, it's not working. Okay. More cytokines, chemokines, call cells. So if this bacteria is something you've been exposed to, all right, in this image, we have basically a lymphocyte that's a B cell with antibodies already produced to that bacteria. We're assuming this person's immune system has seen this bacteria before. The chemical cries for help don't just call the mast cells and other cells like natural killer cells. It calls those lymphocytes, those B cells with antibodies in, and then the B cells chuck that antibody at the pathogen. And then what happens is the pathogens get all caught up with those antibodies, and then they're inclined, all those pathogens, bacterial cells will clump together in an agglutination reaction. When have you seen an agglutination reaction this semester? Blood typing test, okay? The flex that you saw, that's exactly what happens when antibodies that are designed against a specific antigen find that antigen and they stick to it and they cause the antigens to clump together in agglutination. And you saw how fast that happened in a little dish, right? Minute or two. That's how fast it is when your body has seen those antigens before and it has antibodies against them. Okay. So we've gone through basically internal innate immunity, all right? So white blood cells, neutrophils, monocytes. Monocytes get into the tissue through extravasation, become macrophages, encounter a pathogen, consume it, become an APC, present that antigen to a helper T cell. And while all this is happening, the chemical cries for help released by these cells pulls in natural killer cells, mast cells, complement proteins. So that is what's protecting you in the 2 or 3 weeks before you have antibodies to an antigen that you've never seen before. So if you got vaccinated for COVID, that 2 to 3 weeks, this was all put in place. And then about 3 weeks later, you'll have B cells making antibodies to protect you. All right, so all that makes sense? Because that's a lot of stuff going on. So now we're delving into adaptive immunity. That is the holy grail of your immune system, is to have antibodies that provide long-term protection. Long-term, like a year, sometimes a lifetime. It depends. It depends. So adaptive immunity is provided by lymphocytes. Oops, no, it went back. Come on, Ben. Okay, lymphocytes include T lymphocytes, abbreviated simply T cells, and B lymphocytes. The T cells we know from the case study, they're produced in the bone marrow, just like B lymphocytes, but they mature in the thymus for whatever reason, they mature there. All right. They're involved in cell-mediated immunity. Cell-mediated immunity is all T cells. Okay. The helper T cell must be activated in order to activate these. So B cells kind of wait around to be told what to do. Sounds silly, but it's true. You need that activated helper T cell to take over everything that happens in this right here. That's why T cells are the most important part of your long-term specific adaptive immunity. B cells are produced in the bone marrow, but they mature in the lymph nodes and the spleen, and they're the ones that produce the antibodies. Thus, B cells are part of antibody-mediated immunity. Okay, so this is just all vocab, all vocab. Did anyone get any of this information or information similar to it in microbiology? That's good, because we don't necessarily get together and say, I'm covering this, are you covering? It's a happy accident. I'm glad. Okay, so T cells getting activated. That's the important part. So this is your macrophage that has destroyed a pathogen. It's now walking around with the pathogen and it's waiting for the helper T cell to arrive. And this is what goes on. So again, cartoony, because this is just how I happen to see our immune system. That's what I think of. So here we've got a whatever pathogen, blah, right? And here we have a macrophage that's going to consume it, break it down, and then wear that antigen on its surface. So it goes like this. So macrophage eats the bacterial cell. Then part of that bacteria's antigen goes on the cell surface. Now it's an antigen presenting cell. Then the antigen presenting cell says, hey, helper T, here's the antigen. And helper T says, thank you. Now I'm activated. And then we're going to talk about all the things that happen with other T cells and then the B cells. So we're almost to the antibody presenting stage. All right. Okay. So basically, the activated helper T cell takes over everything. It's going to tell other T cells what to do. And we have a total of 4 of them, at least that I know of. And then B cells. We have 2 types of B cells, but the helper T is going to direct the entire event that happens. Okay. So 2 types of adaptive immunity. One that's mediated by T cells. All right. That's cell mediated. And then the other is antibody mediated. That's the B cells. They make the antibodies. So here are all the T cells. The first one right off the top, helper T cell. It is doing everything. So antigen presenting cell presents an antigen to the helper T cell. And then the helper T cell is going to do all the next things I'm going to talk about, but ultimately activate the B cells. So helper T cells now activates other T cells, cytotoxic T cells, T little subscript C. These are also called killer cells, but I'm not going to call them that because that will confuse you, won't you? Because are they the killer cells of adaptive immunity, or are they the killer cells, the natural killer cells that secrete the interferon and the toxic granules? So I don't call it that. I call them cytotoxic T cells because that makes a distinction. So they recognize and destroy a pathogen immediately. So they're just on the prowl, looking for anything that has an antigen they don't recognize, and they're just going to destroy it. Cytotoxic. The other T cell is called a memory T cell. And like the name implies, it keeps a memory of a pathogen. When I say a pathogen, basically the antigen that's on a pathogen, but they keep a memory of that in case they need it later. And then important, since we were talking about an autoimmune disorder for our case study, regulatory T cells, they inhibit cytotoxic T cells and B cells. For every action, there's like an equal and opposite reaction. There's a lot of autoimmune disorders out there. Regulatory T cells are supposed to guard against your immune system from going haywire, kind of keep it in check. That's what they do. And so the leading thought among many things that is going on with the autoimmune disorders that you see is that perhaps regulatory T cells aren't doing their job. And since all T cells mature in the thymus, and if the thymus is overgrown, hyperplasia, that is always indicative of a chronic immune response or a faulty immune response. And it lies with those cells right there. Okay, so 4 T cells, but the most important is right there. Okay, so once helper T cells are activated, you then move on to this. So all these things are happening at the same time. So B cells are activated by the helper T, and they produce antibodies. In nursing, you're going to learn about all the immunoglobulins because you can measure different immunoglobulins in the bloodstream and find out what kind of response is going on in the body. And they're given by Greek symbols like gamma, delta. I can't even memorize them all, but there's a lot of them. Now, once B cells are activated, I need to move this and get this out of my way here. You get 2 types of B cells. You get, come on, pen, there we go. You get plasma B cells. And I'll put here, bless you, ready to fight now. And I'll explain what that means. And you get memory B cells. I'll put can fight later. And I'll try and explain what this means. So when I think of plasma B cells, I think of put up your dukes because you're ready to fight now. They're ready on a moment's notice to defend you. Memory B cells, this is why you get that second booster, or in some cases for certain things like childhood vaccinations, maybe even up to 3. And for COVID, they've gone on and recommended a 3rd, and now they're thinking about a 4th, because what that does is it boosts production of these memory B cells that hang out in your lymph system, your lymph nodes, and wait. They're going to wait and see if plasma B cells put if your duke's ready to fight now, win the war, or find out they're outnumbered, and then all of a sudden you'll have a huge army waiting in the wings to come in. So the memory B cells is the goal of immunization, is you have so many of these cells waiting, and they will come out in force, huge force, and hopefully defeat anything. Okay, so here is your helper T cell. It's going to be activated by an antigen presenting cell. All right, activated. Okay, and then it goes on, activates cytotoxic T cells, memory T cells, regulatory T cells, tells B cells to make their antibodies. And we have two classes of B cells. Okay, so here's what's happening, like I've mentioned. When a white blood cell, like a plasma or a memory B cell, I don't know here, I said plasma, has the antibodies to this particular bacteria, chucks those antibodies out it, and then all those antibodies stick to the bacterial cells. They clump together in an agglutination reaction that looks something like this. All right. Now there's two things going on with agglutination. For one thing, the bacteria are now stuck together. They can't go anywhere. They can't migrate anywhere. This big clump is very obvious to other immune cells in your body, other phagocytic cells, mast cells, natural killer cells. They become conspicuous. They can't hide. They're all stuck together. And so it's like shooting monkeys in a barrel. They can't go anywhere, and other immune cells come in and tear them all apart. And that's one of the functions of agglutination, okay, is to do that and to become very visible to the rest of your immune system. So we saw that agglutination can happen pretty darn quickly in our blood typing test, and that's literally what it looks like, all right, but except inside your body. Okay, so more about B cells. Activated by the helper T, produce antibodies. And here is where, I don't know why there's so many names for it. It's called humoral immunity, antibody-mediated immunity, specific immunity, and I think there's even a couple other terms that call this long-term immunity. So I don't know what they're going to call it when you get into nursing. So I'll throw all the synonyms, similar meaning names at you. Okay, so exposure of the B cell causes the antibodies to be chucked at the pathogen, and then everything gets stuck together with agglutination. Okay. And then something else happens. That's really cool. Once the B cell, whether it's plasma or memory, detects that antigen and chucks its antibodies at it and causes agglutination to happen, that white blood, not white blood cell, that B cell or plasma B or memory B cell clonally replicates itself. So you get one copy, then two copies, then each of those have two copies. Now you're at four. Each of the, it's logarithmic increase in replication of those cells to fight that particular pathogen. Okay, that's not something a lot of people know, is that once that B cell chucks its antibodies at the pathogen, it replicates itself. And now all of a sudden, you've got a whole bunch more of those immediately. Okay, so the clonal production of two types of cell, this is repeat, plasma B cell, I'll just put up your dukes. Is that how Dukes is spelled? I don't know, it's such an old phrase, but I come up with these things and it helps me. They fight now. And memory B cells fight later. But these memory B cells are very important. You will have so much of them. The more you're exposed to something or immunized or exposed, the more memory B cells you're going to have. And so I'm a Lord of the Rings fan, so I have this little thing when we get to that explaining why you do a couple series of vaccinations. So I don't know if I'll get to that today, but this is showing the B cell being exposed to the antigen that it's going to chuck the antibodies. Now they don't show the antibodies being chucked out here, but they show us the clonal replication that happens upon exposure to that antigen. And so this makes your immune system really powerful at defeating pathogens. Here we are. We didn't have the vaccines. We wouldn't be like this today. Without the mask, going out in public again, that's the power of these antibodies in protecting us. The cases I know on campus, because they post it on the university website, have plummeted. I'm not saying it's not out there. I'm saying enough of us have been exposed either to, we've just been directly exposed and have gone through some illness and have recovered, or we've been vaccinated. And now most of us have antibodies against this. And so fewer cases are happening because most people already have the antibodies now. And that's why the cases are low, because this is what happened in our bodies, whether you were naturally exposed or you were immunized. Okay, so effect of the antibodies, well, they activate other immune cells, but the agglutination, that's the big one, okay? And then the clonal replication. So the function of the antibodies is to cause agglutination of the pathogen, which causes other cells to come to the site. And ultimately, exposure to that antigen causes the clonal replication, which protects you long-term and effectively. So this is the cloning, all right? So here's a whatever B cell, plasma, or memory has the antibodies, the little green Y things. Here now we have cocci bacteria, let's say, okay? And then the antibodies bind with that antigen. You're gonna see agglutination, not shown here. But that cell that was exposed to that antigen now clonally replicates itself. About half of those will be more plasma B cells. I'm ready to fight now. And then memory cells, I'll fight later if I need to. I'll be waiting in the wings, watching the whole thing go down. And if I'm needed, I'll be there. Is it coming across okay? I know a lot of it is imagination based because these are microscopic things happening. Okay, so here is our flow diagram. So here, literally, it looks like this. Once a pathogen enters into the body to the formation of antibodies, and then I have a key here. So let's look at the time. We do have the time. Okay, so bacteria enters in from a break in the skin. What are the white blood cells I just told you that, oop, nope, back, oop, forward. Very sensitive. What are the white blood cells I just told you that were in the bloodstream? Neutrophils and monocytes. Okay, step 2. The cell that extravasates from the blood, released into tissue. So it's the monocyte that extravasates, okay, and then enters into the tissue and is now called a macrophage. Okay, step 4. This phagocytic cell in the tissue, which finds a pathogen, kills it, and puts its antigen on its surface. And you can use the acronym. No, before the helper T, the APC, antigen presenting cell. Okay, now from here to here, we have communication between innate immunity and adaptive. So now we have a cell of cell-mediated adaptive immunity, which becomes activated by interaction with the cell above, the APC. That is the helper T. Okay, now, let me get this, it's always in the way. Step 6, activated helper T cell from 500, go back. Activates these. What is a cell of cell-mediated immunity? So you know it's going to be a T cell, which can directly kill the pathogen. It's a T something. Cytotoxic T, I'll put TC, a cytotoxic T cell. Here's another cell of cell-mediated immunity, which retains a memory of the pathogen. The memory T cell. I'll just do that. And now a cell that is part of antibody-mediated adaptive immunity. So the helper T cell activates B cells. That is, when it says antibody-mediated, only the B cells make antibodies. Now, the B cell make these things, which are known as immunoglobulins, antibodies. Now, the B cell from that above encounters this pathogen, and the following happens. So it's got antibodies. It's going to chuck those antibodies at the pathogen and cause them to stick together. What's that called? Agglutination. Okay, and in that, the antibodies stick to the pathogen. And then once that B cell has come in contact with the antigen, it's going to make more of itself, clonal replication. Oop. And that includes both the plasma, where'd my L go? And memory, B cells. Okay, so that's it. Bacteria gets in, the sequence, a very simplified, yes, 2 antibodies, and then what those antibodies cause to happen, 2 important processes: agglutinate the pathogen, and then other cells come in and destroy it, and then clonally replicate more of those cells for that antigen. There's your immune process. But this takes 2 to 3 weeks. All right, 2 to 3 weeks for that to happen. Okay, so again, there's going to be a question on the final exam of this. I'll have all these sequences numbered with different numbers and all mixed up. And then you have to be able to go through step 1, step 2, step 3. Okay. Here's another way of studying the entire immune system. All right. I think we'll be able to finish this after this on Friday. So innate immunity is called, go back, non-specific. Adaptive immunity is known by many things. It's called specific or humoral immunity. I'll just put immune, period. Okay. We have external innate, and we have internal innate. Externals are barriers. Give me a barrier that we have to the entry of a pathogen. Skin? Yep. What else? Where can things get in? Tonsils. That's airway. Okay. Also, now I don't know if I have all the right bulleted points that match with the key I'll look at. Alveolar macrophages. I just can't fit it in. What's another possible entry point? Eyes, tears are antimicrobial. What happens if you consume it? Stomach acid. What happens if it tries to get into the vagina or urethra? Okay, are barriers. What happens if it gets in? Starts here. White blood cells, neutrophils, monocytes. They encounter a pathogen in the bloodstream. What can they secrete that causes a fever? Oh, that's cool. I don't necessarily want that. No, I don't want that. Go back. It's not letting me. Come on. Really? Escape. Here we go. I'll just put pyro G because I can't fit it all in. What are the chemical cries for help? Cytokines, chemokines. Okay. The monocyte extravasates, now it's in the tissue, and it's called another M word, macrophage. And if it encounters its pathogen and puts that antigen on its surface, it becomes an APC. Okay. Then we have the cells that secrete interferon to kill viruses or toxic granules to kill tumor cells that are the natural killer cells. What are the cells that secrete histamine? Mast cells. It's hard to write on this tablet when the screen's folded like this. And what are the things that poke holes in bacteria and lyse them? Complement proteins. Okay. We have cell-mediated here. Cell-mediated is all T cells, right? What's the most important T cell? Helper T. And then what's the T cell that can kill directly? I'll just put TC because I don't think I can fit cytotoxic in there. What's the one that has the memory of a pathogen? TM. And then what's the T cells that suppress B cells and cytotoxic T cells to slow your immune system down? Regulatory. Okay. Then we have antibody mediated, and we're almost done because this is the entire PowerPoint. We have two types of B cells. What are the ones that put up your dukes, you're ready to fight now? Plasma B. And then the one that waits, memory B. There it is. All right. So 2 ways to start the entire immune system. And we're going to pick up the rest of this chapter on Friday and finish it. And so that means next week, we'll start reproductive physiology. Yay, my favorite. But we'll be able to get this done. All right.